Very good morning, all the way from New York. My name is Dr. Tenzin, and we had the privilege of having a lot of audience all over India. I know, by the way, good evening, India. I know you guys are watching your YouTube live from India. So for the last 2 days, we're having the NEET series for the exams. I know that all of you guys are really scared about the exams coming up in January. So for the last 2 days, we had two great subject experts teaching anatomy and pharmacology. So today we have Dr. Karthikeyan, who is an MD in biochemistry. He's a leading faculty of the subject, very popular amongst his students in MAMC and AIMS for his clarity and student-friendly method of teaching. Known for his very strong grip on the subject, he is also very actively engaged in teaching students in virtual platforms and social medias like WhatsApp and Facebook. He has authored and contributed to many exam-oriented biochemistry books, and recently he has been acknowledged in the 31st editions of Harper's Illustrated Biochemistry textbooks for his suggestions and corrections. He is currently pursuing his PhD, studying the epigenetic changes in AML under the mentorship of Dr. Subrati. So ladies and gentlemen, without wasting much of your time, I will give you the best and the most the experienced biochemistry tutor or teacher or professor. We have Dr. Karthik.
Thank you, Dr. Tenzin, for the introduction. Dear friends, welcome you all to this live 1-hour session on biochemistry. So let us utilize this session to the most. I have designed this session in such a way that we will get everything. First, I will tell you some useful mnemonics. Then I will clear certain doubts students ask me every time through WhatsApp and Facebook and all. Then we will quickly review some important concepts and some image-based questions and discussion also. So you can ask me questions in between. I will try to answer. So let's begin. Mnemonics are very, very important. For example, this is very important. NADPH producing reactions. There are just 3 reactions that produces NADPH. That is G6PD of HMP shunt, malic enzyme, and cytosolic isocitrate dehydrogenase. Okay, so they can easily ask all of the following reactions produce NADPH except, okay, they can just give 4th enzyme. For example, they can give malate dehydrogenase. Okay, many students confuse malic enzyme and malate dehydrogenase. Both are different. Malic enzyme converts malate to pyruvate. Okay, so it is involved in oxidative decarboxylation, whereas malate dehydrogenase converts malate to oxaloacetate. Okay, so how will you remember this? Just remember as Goa Medical College or Government Medical College. G6PD, malic enzyme, cytosolic isocitrate dehydrogenase, right? So the question can be like this. So the answer will be what? Malate dehydrogenase. 5 cofactors for PDH. This is very, very important. Pyruvate dehydrogenase requires 5 cofactors. These can be memorized as tender, lovely, care for Nancy. Okay, tender, lovely, care for Nancy. T, thymine pyrophosphate. L, lipoic acid. C, coenzyme A. F, FAD. N, NAD. Okay, so here if you see that vitamin B1, B2, B3, B5. So all these 4 vitamins are required for PDH. So this can be also a question. Okay, all of the following vitamins are required for pyruvate dehydrogenase except, so they can easily ask, right? So tender, lovely, care for Nancy. Don't forget Nancy, right? The order is also important because this is the order in which electrons are transferred in the enzyme. So PDH transfers electrons in this order. So the final acceptor of electrons in the PDH complex is NAD. The final acceptor of electrons in the electron transport chain is your oxygen. Okay, so oxygen is the final acceptor of electrons in electron transport chain. Okay, now how will you remember FADH2 releasing reactions? There are a few reactions which convert FAD to FADH2. So these are fatty acyl CoA dehydrogenase, glycerol 3-phosphate dehydrogenase, which is in the mitochondria, succinate dehydrogenase. So you can remember this like foreign graduate students. Okay. So in many colleges nowadays, we have foreign graduate students also. So remember like foreign graduate students. So let me tell you, you should always remember those things in which you have very few points. For example, FADH2 releasing reactions, you have just 3 reactions. Don't try to remember those reactions in which NAD is involved. There are a lot of reactions in which you require NAD plus. But FADH2 releasing reactions are very few. These 3 only. Okay. The sequence of TCA cycle can be easily remembered like this. Can I ask some sweets for mom? Okay. So remember like this. Clear? Next, glycogen storage disease. This is high yield topic, right? So this can be remembered like very personal chats are mostly hidden. Okay. So type 1 is Van Gerkes, type 2 Pompe, type 3 Cory's disease, 4 Anderson, McArdle's and hers. Okay. So here again, let me tell you, how will you remember the enzyme deficiency? For right? So it is muscle phosphorylase. H for H. So it is hepatic phosphorylase. So this is muscle glycogen phosphorylase. His disease is hepatic glycogen phosphorylase. Again, see here. CDAB. So remember like this, ABCD. Anderson disease is branching enzyme defect. Cory's is debranching. It's fun, right? A, B, C, D. Pompe's disease, everyone know it is lysosomal acid maltase defect. Van Gierk's, you should know it is glucose 6 phosphatase defect. So Van Gierk's disease is a defect of both gluconeogenesis as well as glycogenolysis, right? Other glycogen storage disorders like this one is important. Fanconi-Bickel disease is due to defect in the GLUT2. Okay, so because of GLUT2 defect, the liver cannot store glycogen. Okay. Essential amino acids, people have this mnemonic AV Hill MPTT. So that is also perfect. Let me give you another way to remember them. It's very easy. You can remember as all the 3 branch chain amino acids, all the 3 basic amino acids, 2 aromatic amino acids, and 2 amino acids that end with ine. So in this way, you can also remember what are the branch chain amino acids, what are the basic amino acids like that. Okay. So what are the branch chain amino acids? Leucine, isoleucine, valine, live amino acid, right? HAL, histidine, arginine, lysine. These are the basic amino acids. And phenylalanine, tryptophan, these 2 aromatic amino acids are essential, whereas tyrosine is not essential. Why? Because tyrosine can be synthesized from phenylalanine by phenylalanine hydroxylase reaction. But sometimes tyrosine can become essential when in phenylketonuria. In phenylketonuria, you have defect in the conversion of phenylalanine to tyrosine. So tyrosine becomes essential amino acid in phenylketonuria, right? Threonine and methionine. So these are the amino acids that end with ine, right? So threonine, methionine. So this is how you can remember the essential amino acid. You can use the mnemonic AV Hill MPTT or this one. Now, there are amino acids which donate one carbon group. So what are they? They can be remembered like this: GST, goods and service tax. So GST is very popular, right? So GSTHM. So GST by Honorable Minister. Okay, so use this mnemonic: glycine, serine, tryptophan, histidine, and methionine. So these amino acids donate one carbon group. So this is a high yield mnemonic. Stop codons: UAA, UGA, UAG. Okay, and see this. These are the names for stop codon. So they can just ask you which is amber codon. Then it will be UAG. Okay, how to remember the stop codons? It's a fun mnemonic. You are away, you gone away, you are gone. That means these do not code for any amino acid. Let's say, okay, stop the translation, go. So that's why you are away, you gone away, you are gone. So out of this, which codon codes for selenocysteine? The answer is UGA. Selenocysteine is the 21st amino acid which is coded by a stop codon, UGA. Right? Now, this is again important, epimers of glucose. So epimers of glucose can be remembered by this: Maggie. What are epimers? Epimers are isomers which differ in the configuration of just one carbon. Okay, if you see the all other carbons, the stereoisomeric pattern is same. The attachment of H and OH is same except in a single carbon. Okay, so the difference between glucose and mannose is just the second carbon. Okay, the difference between galactose and glucose is just in the fourth carbon. So mannose is the C2 epimer, galactose is the C4 epimer of glucose. Whereas mannose and galactose are not epimers of each other. Okay, this is very, very important. Galactose is not the epimer of mannose. They are just stereoisomers, right? So this allose and idose, they have been asked anytime, but it's very easy to remember, right? Remember Maggie. Okay, so start from 2, 2, 3, 4, 5. So mannose is the C2, galactose is the C4 epimer. Vitamin K dependent clotting factors. Vitamin K is involved in the gamma carboxylation of clotting factors. So how will you remember the numbers? So now you orient your anatomy knowledge. In anatomy, you might have studied that factor 3, 7, 9, 10. Sorry, the cranial nerves 3, 7, 9, 10 are the cranial nerves carrying the parasympathetic supply. So with this, now you can integrate biochemistry. Vitamin K dependent clotting factors are 2, 7, 9, 10. So cranial nerves carrying parasympathetic supply 3, 7, 9, 10. Whereas vitamin K dependent clotting factors are 2, 7, 9, 10. Okay. Not only clotting factors, but also other proteins require vitamin K for the gamma carboxylation. So they are protein C, protein S, protein Z, and even your bone forming proteins. Okay, so that is why chronic warfarin therapy leads to osteoporosis. Okay, remember this. Inhibitors of translation can be remembered by this famous mnemonic: buy at 30, sell at 50. So A, aminoglycosides, T, tetracycline. So buy at 30, right? So these two are the inhibitors of 30S. You know, 30S ribosome is involved in the recognition portion. Okay. So the large 50S ribosome is involved in the actual peptide bond formation. So this is inhibited by cell at 50. So it's a very famous mnemonic everyone knows. So chloramphenicol, erythromycin, linezolid, and lincomycin. Okay. Now, it's a very interesting mnemonic. See, in South, people eat Dosa, right? So south is dosa. In north, people eat roti, okay, chapatis. In west, people eat pizza, okay. So southern blot is for DNA, northern blot is for RNA, western blot is for protein, okay. So what is southwestern blot? You compare these two, combine these two. It is DNA protein interaction, okay. So someone is asking, vitamin K dependent factor, is it osteopontin or is it osteocalcin? Okay, so which is that factor? It is actually osteocalcin. Okay, so osteocalcin is the factor which require vitamin K. So if you look at the osteocalcin structure, it requires gamma carboxyglutamate. Why we need that gamma carboxyglutamate? Because calcium has to bind. See, look at this. This I have. Let me show in this. See, glutamate contains only one carboxyl group in the gamma portion. Okay. Calcium is a divalent cation. So for it to bind, we have to provide two negative charges, right? So that two negative charges are provided by your gamma carboxy glutamate. Okay, so it is 2, 7, 9, 10 protein C, protein Z, and osteocalcin. Okay, it is not osteopontin. Remember this. So these are the mnemonics I have told you for today. Okay, so I always suggest you to have your own mnemonic. Okay, so you will never forget your own mnemonic. Okay, and use those mnemonics that are spicy. Okay, add some spices in the mnemonic. Okay, use your own mnemonic. I know you understand what is the meaning of spice, right? So add spice in the mnemonic, you will remember. Okay, so with this, let's move on to the next section, doubt busters. I get these doubts many times in my WhatsApp, Facebook, and all. So these are those doubts. A few of the doubts I'm explaining because we are in short of time. First doubt many people ask is tyrosine polar or not polar? Okay, so tyrosine, it contains what? This ring, phenol ring, right? So it contains OH group. That does not make it polar. Okay, it has this bulky aromatic group. Because of this bulky aromatic group, tyrosine is non-polar. Clear? Hmm? But still, when you compare among the other non-polar amino acid, it is the least non-polar. Again, is glycine polar? People ask me. Glycine is the smallest amino acid. The R group of glycine is just hydrogen. It is optically inactive amino acid. There is no D form, L form for glycine. It is the smallest. The R group is hydrogen, so it is polar. But it is the least polar among the polar amino acid. Okay. So take it from me, the most polar amino acid is arginine. Arginine contains delta guanidino group. Okay. The most non-polar amino acid, if they ask, it is your isoleucine. Okay. Which is the purely ketogenic amino acid? See, in NEET, you have just only one choice. So the answer is leucine. Okay. So certain textbooks say lysine is also ketogenic, but for NEET exam purpose, go for leucine, right? This question, this is a highly controversial question because students make it as a controversy. There is nothing about controversy. So how many ATPs are required for formation of one peptide bond? So this is the doubt. The answer is 4 high energy phosphate bonds. Otherwise 4 ATP equivalence. So ATP equivalent, that means GTP is a ATP equivalent. Hydrolysis of GTP to GDP is 1 ATP equivalent. Okay. Hydrolysis of ATP to AMP plus PPI, that is inorganic phosphate, this is actually 2 ATP equivalent. Remember this. When ATP is converted into AMP, we call it as 2 ATP equivalents. Okay, the 1 ATP equivalent is nothing but one high energy phosphate bond. So you might have studied that 4 high energy phosphate bonds is required for the formation of peptide bond overall. Okay. But in the exam, if they ask you like this, how many ATPs are required for peptidyl transferase reaction? So the peptidyl transferase is the ribozyme. So what is this? This is a ribozyme. 28 is ribosomal RNA. So this ribosomal RNA does not require any ATP. Okay, the formation of peptide bond by this enzyme does not require energy. That does not mean that peptide bond formation does not require energy. Okay, so if they specifically ask you that by this enzyme peptidyl transferase, then only you have to write the 0. Otherwise, it is 4 ATP equivalent. Okay. Narayan is asking about easy way for porphyria. Okay, so Narayan, I hope you will find it in the internet. So we will discuss this in some other time. And this is again a highly controversial thing. Many students confuse this Warburg effect. Okay, so Warburg effect is aerobic glycolysis. Okay, so remember this Warburg effect is aerobic glycolysis. What is aerobic glycolysis? It is nothing but glycolysis in the presence of oxygen. Okay, even in the presence of oxygen, glycolysis is happening. Okay, so actually glycolysis is nothing but conversion of glucose to pyruvate. Okay, so that is actually the glycolysis. So here, even in the presence of oxygen, glucose is converted into pyruvate. As glycolysis rate is high, pyruvate is converted into lactate. Why? NADH is important for the progression of glycolysis. So to regenerate NAD plus, okay, it's NAD plus. So NADH needs to be converted into NAD plus. So free NAD plus is important for the progression of glycolysis. So to give the free NAD plus, this conversion of pyruvate to lactate is happening. Okay, so Warburg effect is aerobic glycolysis. It is the conversion of glucose to lactate. Okay, it's not lactose. In your Robbins textbook, it's wrongly given as lactose. It is actually conversion of glucose to lactate even in the presence of oxygen. Clear? Now, this question, rate limiting enzyme of glycolysis. So people say, why can't it be hexokinase? So the rate limiting enzyme is the enzyme which catalyzes the slowest possible step of the reaction, and the product should be unique. Okay, if you see, glucose is converted into glucose 6-phosphate by hexokinase, but glucose 6-phosphate can go to other pathways like HMP shunt and all. So glucose 6-phosphate is not unique to glycolysis. Okay, glucose 6-phosphate has various other fate. It can go to glycogen synthesis and all. Okay, so that is why hexokinase is not the rate limiting enzyme. It is the phosphofructokinase, that is formation of fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate has no other fate other than proceeding in the glycolysis. Right? Now, this question, flux generating enzyme of glycolysis. This is your hexokinase. Okay, so hexokinase converts as much as glucose into glucose 6 phosphate. So it creates the flux. So it puts the glucose 6 phosphate into the glycolysis. So flux is the rate of reaction. So hexokinase put as much as possible glucose into glucose 6 phosphate. Okay, so hexokinase has the less affinity for glucose. Even in the lowest concentration, glucose can be converted into glucose 6 phosphate. That's why we say hexokinase is the flux generating enzyme of glycolysis. This question, see, how many ATPs are produced from conversion of fructose to lactate? Okay, so people may think that, okay, conversion of fructose to lactate, we get one more ATP and all. No, it is the same as that of glycolysis. You get 2 ATPs. Okay, so why? The reason is, see, fructose, if you phosphorylate fructose, you get what? From fructokinase reaction, you get fructose 1 phosphate. Okay. Now, if you see, you have just fructose 1 phosphate. Okay. So you used 1 ATP here. Clear? Closely follow me. See, you used 1 ATP here. Now you break this fructose 1 phosphate by the aldolase catalyzed reaction. You get dihydroxyacetone phosphate. Okay, you have dihydroxyacetone phosphate, which can go into glycolysis. But what you get glyceraldehyde, right? So we have to phosphorylate this glyceraldehyde also. So we use another ATP. So it is like glycolysis only, right? So in glycolysis also, we have to use 2 ATPs. Okay, so we get 4 ATPs. So 4 minus 2, you get 2 ATPs. It is the same case with fructose also. Okay, so don't get confused. The energy yield of glucose to lactate and fructose to lactate is same. Clear? I hope you all got my point. Now, this is the time for quick review. Dear friends, we have crossed half an hour session. So we have half an hour more. Let us spend this time in a useful way. Let's utilize this half an hour 100%. So let me quickly review certain important concepts again. Students confuse oxidative decarboxylation. See, what is oxidation? It is loss of electron, removal of electron. Decarboxylation is removal of carbon dioxides. Okay, so what are the oxidative decarboxylation reaction? Pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, isocitrate dehydrogenase, branched chain keto acid dehydrogenase. Okay, here again, among these, one enzyme does not require vitamin B1. What is that enzyme? Can anyone tell me? Tell me in the comment section. Which enzyme does not require vitamin B1 among these? Yeah, correct. It is isocitrate dehydrogenase. B1 is required for other three, not for isocitrate dehydrogenase. Okay. So don't think that vitamin B1 is important for all the oxidative decarboxylation reactions. Okay. It is required for only these three oxidative decarboxylation reactions. Okay. Then there is something known as oxidative deamination. Again, oxidation is loss of electron. So here the electrons are removed in the form of either NAD or NADPH. Another high yield point. Glutamate dehydrogenase is the only enzyme which can use both NAD as well as NADP. Okay, so it can use NAD as well as NADP. Okay, so this enzyme can use both. The NADPH is used in the reverse reaction. In the forward reaction, NAD plus is used. It's not required for you. For you, you should know that it can use both. Okay, that's all. So decarboxylation is removal of carbon dioxide. So glutamate is converted into alpha ketoglutarate. Okay, again, what is the detoxification form of ammonia? Okay, this is again a favorite question for examiners. So if it is all over the body, okay, all over the body, the detoxification form of ammonia is glutamate. Okay, glutamate transamination, right? If it is brine, ammonia is toxic to brain. Why? Excess ammonia causes depletion of the intermediates of TCA cycle. Why? Alpha ketoglutarate is converted into glutamate. So alpha ketoglutarate is important for TCA cycle. Without alpha ketoglutarate, you won't get any ATP. So the brain takes one more extra step. It converts glutamate to glutamine also. Okay. So that is why from brain, it is glutamine. Okay. If they ask you about muscle, okay, in muscle what happens? Pyruvate is converted into alanine. So alanine is the detoxified form. So this is very, very important. Okay, you should know this. All over body, glutamate, brain, glutamine. From muscle, you have alanine. Clear? So oxidative deamination reaction is catalyzed by glutamate dehydrogenase. Where is this reaction taking place? It is in the hepatic mitochondria, right? Now, I told you to remember these enzymes, which are very few. For example, only 3 enzymes require B12. Out of that, only these two are in the human body: methylmalonyl CoA mutase and methionine synthase. Okay, only these two are the human enzymes that require B12. Leucine amino mutase requires B12, but it is not present in humans. Okay, see this. From this, they can ask one question. How will you treat methylmalonic aciduria? Yeah, acidemia. Which vitamin will you use? See, this methylmalonyl CoA mutase is required for vitamin, is required B12, right? So we can treat methylmalonic acidemia by treatment of cobalamin. Okay, this is very, very important. And one more question they can ask: how will you diagnose B12 deficiency? They can give methylmalonic aciduria. So presence of methylmalonic acid in the urine is one of the diagnostic tests for B12 deficiency. Clear? Again, I told you only 4 enzymes require: vitamin B1, PDH, alpha ketoglutarate, and BCKD. It is branch in keto acid. It is not branch in amino acid dehydrogenase. Remember, it is branch in keto acid dehydrogenase. Then it is transketolase. That is why for B1 deficiency, we assess that increased transketolase activity after addition of thymine pyrophosphate. So in our BCs, once you add thymine pyrophosphate, we can see increased transketolase activity. Otherwise, the transketolase activity is decreased in B1 deficiency. Clear? Okay. Someone is asking me about this one, about non-oxidative deamination. See, I told you about oxidative deamination. This slide, it is saying oxidative deamination. There are certain reactions which are non-oxidative deamination. So what are these non-oxidative deamination reaction? Serine dehydratase, dehydratase. Okay. Threonine dehydratase. Remember this. Serine hydratase and threonine dehydratase are examples for non-oxidative deamination. Without oxidation, we remove the ammonia. Clear? Again, only 4 enzymes require biotin. So it's good to remember the 4, right? Acetyl-CoA carboxylase, pyruvate carboxylase, propionyl-CoA carboxylase, and methyl-crotonyl-CoA carboxylase. See this 4th one, many of you might not have studied about this 4th enzyme. Please look at this. It is methyl-crotonyl-CoA carboxylase. It is involved in the metabolism of amino acid. Okay, branch in amino acid metabolism. You have this. And many people think that all amino acids require, all carboxylases require biotin. No, it's not true. Okay, there are biotin independent carboxylases. These are the carboxylase enzymes that does not require biotin. It's like addition of C6 to the purine ring. It is done by amino imidazole ribonucleotide carboxylase. Remember as AIR, All India Radio. AIR carboxylase. Okay, formation of carbamyl phosphate by the enzyme carbamyl phosphate synthetase 1 and 2. That does not require biotin. And as we have already seen, vitamin K mediated gamma carboxylation of clotting factors and other proteins also does not require biotin. Okay, so this has been already asked in your previous exam. They can ask these two. Okay, AIR carboxylase and carbamyl phosphates, that is 1 and 2. Remember, it's very, very important. Okay, so someone is asking P bad requires B1. So what do you mean by P bad? So can you P bad? Oh, he's giving a mnemonic. Good, great, great, great. He's giving us a mnemonic. It's like pyruvate, branch chain, B, A, alpha ketoglutarate, T, transketolase. Great. See, this is how. See, he created a mnemonic, right? From this. Good. Thank you. You all getting benefit from his mnemonic. Thank you. Now, this topic, epigenetics. Uh, this has been asked many times, many a times. So let me tell something about epigenetics. Epigenetics refers to heritable change in gene expression without change in underlying nucleotide sequence. See, usually when we say that when gene expression changes, okay, so when gene expression change, so it changes when the DNA sequence change. So GAG, when GAG is converted into UAG, you are gone. It's a stop codon, right? So just stop codon. So now the gene expression will change. Okay. But sometimes without any change in the underlying nucleotide sequence, the gene expression can be altered and it is heritable also. So these heritable change in gene expression without change in the underlying nucleotide sequence is known as epigenetics. So how will you change the gene expression without changing the nucleotide sequence? So either it can be done by putting a methyl group in the cytosine. Okay, methylation of cytosine of DNA. Once you put methyl group in the cytosine of DNA, that causes suppression of gene expression. Okay, repression. You can use the word repression or suppression of gene expression. There is another word. It is like heterochromatic formation. That means the DNA is tightly bound by proteins. It is not expressed. Okay. Now, this is one way. Either you put a methyl group in the cytosine of DNA, or you know that the negatively charged deoxyribonucleic acid DNA is bound by the positively charged histone proteins, right? So now you remove the positive charge of the histone proteins. Okay, you put acetyl group. Acetyl group is negatively charged, right? So acetylation of epsilon amino group of lysine. Okay, this is the amino acid. Lysine is positively charged. So this is responsible for the positive charge of the histone protein. Now you mask the positive charge of this. So acetylation of lysine of histone will favor euchromatin formation. That means it will open up the histone. Now the DNA is accessible to your gene expression machinery. It is accessible to DNA polymerase, RNA polymerase. So now it can be converted into mRNA, messenger RNA, right? So this is epigenetics. So in epigenetics, they can just ask about the definition, or their favorite question is methylation of cytosine. Which residue? Once they have asked which residue is methylated, here you can put cytosine. Okay, methylation of cytosine. That is gene repression. Then acetylation of which amino acid? Lysine. Which part of lysine? It is the epsilon amino group of lysine. It is the epsilon amino group. What it will do? It will favor the gene expression. So otherwise you can also say euchromatin. Okay. So these are the usual questions they ask about epigenetics. Okay. I always suggest you don't go very deep into any topic. For example, one student was studying everything about CRISPR-Cas9 system. Okay. CRISPR-Cas9 system. What I will suggest you is, okay, don't go into very deep. You have so many subjects to study. All you have to know about CRISPR-Cas9 system is it is a gene editing tool. Okay, we use this to edit genes. So earlier days, we use other systems also. Okay, so we used to use Cree-Lox system. Okay, just know this name, Cree-Lox system, Cree-Lox-P. Okay, Cree-Lox system also we used to use. But this CRISPR is better than the other gene editing tools. Okay, you might have studied in newspapers. Okay, in China, some scientist, he developed baby resistant to HIV infection by using this CRISPR-Cas9 system. The advantage of CRISPR-Cas9 system is it is very accurate. Okay, so you can exactly target, you can exactly change the sequence. Okay, so just you should know that it is a gene editing tool, right? How it works, it based upon the DNA repair mechanism. Okay, I will give you one extra point. So it based on the DNA repair mechanism, we actually give, we can give, it can work on non-homologous, homologous repair. It's both the way. Say, don't study what is non-homologous until now. Okay, it can work on non-homologous DNA repair, double strand break repair, or homologous repair, or homology directed repair. Okay, all you have to know is it based on the DNA, double standard DNA repair mechanism. So that is the mechanism of CRISPR-Cas9. We give the DNA strand itself, so that based on that, we cut one sequence and we will put another sequence, the desirable sequence. So this can be like, it can be used for gene therapy. So you remove the mutated gene, you put the desirable gene. Okay, so CRISPR-Cas9, that means it's a gene editing tool which works precisely. Okay, that is the important point. It precisely cuts. Now, we have many more things to study. We have just 19 minutes left with us. Let's use this time effectively. Rate limiting enzymes. Okay, you will always find this in any books. So always go through this. Glycolysis, phosphofructokinase 1. Glycogen synthesis, it is glycogen synthase. Glycogenolysis, glycogen phosphorylase. Now let me ask you one question. Which is the enzyme which is common to both glycogen synthesis and glycogenolysis? Which is that enzyme? It is what? Phosphoglucomutase. Okay, so they are, these days they are fond of these kind of question. Which is the enzyme common to both glycogen synthesis and glycogenolysis? The answer is phosphoglucomutase. Which is the enzyme common to both glycogenolysis and gluconeogenesis? The answer is glucose 6-phosphatase, right? Glucose 6-phosphatase is a marker of endoplasmic reticulum. Beta oxidation of fatty acids. The rate limiting enzyme is CPT1, which is inhibited by malonyl-CoA. Malonyl-CoA is the substrate for fatty acid synthesis. So that is why during fatty acid synthesis, fatty acid oxidation is inhibited, right? Fatty acid synthesis, the rate limiting enzyme is acetyl-CoA carboxylase. I told you acetyl-CoA is converted into malonyl-CoA. This in turn inhibits CPT1. They can ask you, which is the allosteric activator of acetyl-CoA carboxylase? Acetyl-CoA is in the monomeric, acetyl-CoA carboxylase is in the monomeric form. To be active, it has to be converted into a polymeric form. So for this, we require citrate. Okay, citrate favors the polymerization of acetyl-CoA carboxylase. Okay, ketone body synthesis, it is HMG-CoA synthase. Okay, why it is not HMG-CoA lyase? You can read in your books. De novo purine synthesis, it is PRP. Synthetase is the rate limiting. This enzyme, aminotransferase catalysis, is the committed step. Cholesterol synthesis, everyone knows it is HMG-CoA. Reductase, it is not HMG-CoA synthase, it is reductase, which is inhibited by statins. And these are the other list also. Okay, so bile acid synthesis. So this they can ask 7-alpha hydroxylase, polyamine synthesis, ornithine decarboxylase. So you remember this, you can answer a different question. See, they can ask in exam, which of the following amino acid is the precursor of polyamines? If you know that the rate limiting enzyme is ornithine decarboxylase, so in the question, if ornithine is there, then it is the answer. Okay. In a rare situation, there is no ornithine. So instead of ornithine, arginine is there. Okay, then arginine is the answer. Why? You know, urea cycle, go to urea cycle. From arginine you remove the uh, you remove urea, right? So from arginine you remove arginase, you use what? Arginase. What you get? You get ornithine, right? So ornithine can be produced from arginine. So the precursor of polyamine synthesis, that it can be either ornithine or arginine. What are the polyamines? Putrescine, spermidine, and spermine. Okay, these are the polyamines. They are very, very important for cell cycle regulation, cell division, etc., etc. And analog of fluorinated analog of ornithine is used in trypanosomal disease also. Okay, and this is very, very important. These are the named cycles and named pathways. See, you know about glucose alanine cycle, but it is known as Cahill cycle. For NEET entrance examination, it's very important to know about these things. Cahill cycle is glucose alanine cycle. Cori cycle, everyone know. Glucose is converted into lactate in RBC and exercising skeletal muscle, and now lactate is converted into glucose back in liver by gluconeogenesis. The other name of glycolysis is Embden-Meyerhof pathway. Sometimes we forget. So they can just ask, which is the rate limiting enzyme of Embden-Meyerhof pathway? Then you are like, what is this Embden-Meyerhof pathway? Okay. And see, look at this. Krebs-Hanslade cycle. This is the other name of urea cycle. It is the Krebs-Hanslade cycle. Okay. Breakdown of galactose is also known as Lillard pathway. Okay. And Meester cycle, this is very, very important. Meester cycle is glutathione mediated absorption of neutral amino acid. And another very, very important one is Rappaport-Lubering shunt. Okay, Rappaport-Lubering shunt happens only in the RBCs. Okay, where that too in the when you want to unload the oxygen. Okay, so Rappaport-Lubering shunt produces 2,3-bisphosphoglyceric acid, 2,3-bisphosphoglyceric acid stabilizes the T state of taut form of hemoglobin. So oxygen is unloaded from the hemoglobin. It is involved in unloading of oxygen. Okay, it causes shift of the oxygen dissociation curve to the right shift, right? So this is the one. Now, named molecules. Certain molecules are named. For example, clinofragment. Okay, this is their favorite question. They are asking in names. They are asking NEET also. In JIPMER also they ask. So clinofragment is the DNA polymerase 1. It is the Escherichia coli, prokaryotic DNA polymerase 1 without the 5' 3' exonuclease activity. Okay, let me tell you fast that 3', 5' exonuclease activity is required for proofreading. Okay, 5', 3' exonuclease activity is required for DNA repair and removal of primers, removal of RNA primers. So 3', 5' exonuclease means proofreading. 5', 3' exonuclease activity is removal of RNA primers as well as in DNA repair. Roseman fold. Roseman fold is the NADP binding domain of certain dehydrogenase, either NADP or NAD. Okay. So alcohol dehydrogenase, your alcohol dehydrogenase, lactate dehydrogenase, these all contain this Roseman fold because they require NAD. Sanger reagent, Edman reagent, I hope you know Sanger reagent is phenyl isothiocyanate, PITC. Edman reagent is 1-fluoro-2,4-dihydrobenzene. Warburg yellow enzyme is riboflavin. Okay, flavus means yellow color, right? Flavin. So it is yellow color. Cori ester. This they may not ask, but still it's better to know. Glucose 1-phosphate is also known as Cori's ester. Okay, and these are some named chemical reaction. Lohmann reaction is this one. This is creatine phosphate is converted into creatine and ADP is phosphorylated to ATP. So this is kind of substrate level phosphorylation reaction. Okay, so this creatine phosphate is the phosphagen in the vertebrate animals. Okay, so this you have to know in the vertebrates. In invertebrates, it is arginine phosphate, not creatine phosphate. Okay, creatine phosphate is the instant source of energy in a sprint, in 100-meter sprint. So in 100-meter sprint, creatine phosphate will be converted into ATP, but it will be just for the initial 4 to 5 seconds. Okay, so after that, if they ask after 4 to 5 seconds of in a sprint, 100-meter sprint, it is anaerobic glycolysis. Okay, remember this, in a sprint, 100-meter sprint, quickly, it's anaerobic glycolysis. You have to fast, there is no oxygen. But in a marathon, so in a marathon, it is a slow paced. So in marathon, everything it is like aerobic, it is oxidative metabolism. Okay, in marathon, it is like breakdown of glycogen, it's the fatty acids, everything is utilized in the aerobic way. Okay, but in 100-meter sprint, it is anaerobic. Fenton reaction, you should know that ferrous is converted into ferric, but here H2O2 as such is not a free radical. H2O2 is not a free radical, but it is converted uh, into what? This radical, hydroxyl radical. Okay, remember this. So someone is asking about Sanger reagent. Okay, so here I want you to know two things. There is Sanger's method of DNA sequencing. There we use dideoxy NTPs. Okay, so Sanger's reagent, Edman reagent, we have two. So it is Sanger reagent is, yeah, correct. So she mentioned it correctly. It is Sanger reagent is this one. Edman reagent is PITC. Okay, so Sanger's is FDNB. Edman reagent is phenyl isothiocyanate. So Karisma Shahu, you have a very keen eyes. Good luck. Next, Fenton reaction. So Fenton reaction is responsible for the toxicity of iron. So when you have free iron, when iron accumulates, the free iron will convert H2O2 into your toxic hydroxyl radical. This one, the one where dot is there, that is the radical. Okay, hydroxyl radical is the most potent free radical. Okay, so in exam they can ask so many radical among the hydroxyl, among the oxygen radical, among the oxygen. Remember, among the oxygen radical, it is the hydroxyl radical which is most toxic. Okay, someone is asks, please tell me at the last that should study by making questions from books or by ourselves, from lines of books, not any. Okay, I will tell you at the last. We have just 7 minutes. Let's wind up. This reaction, Maillard reaction. Okay, Maillard reaction is the reaction between amino acids and reducing sugars. So this leads to formation of glycation, advanced glycation end products, HbA1c. Okay. Okay, in HbA1c, I got remembered this point. HbA1c, in the latest edition of textbook, if you see, it's like it's not just valine, it is also lysine. Okay, so here the in HbA1c, glucose is attached to valine as well as lysine. Remember this epsilon amino group of lysine and the amino terminal end of valine. So in both glucose is attached. If you see the previous editions of the textbook, there may be just one. Okay, now it is both the two. That is very important. And this can be most abundant. I told you, right? Most abundant amino acid is glutamine. Okay, from the brain it carries ammonia. This thing about collagen is very, very important. The most common type of collagen, most abundant collagen in the body is type 1. But in the basement membrane, if they ask, it is type 4. Most abundant collagen in the cartilage, it is type 2. But in the white fibrocartilage, again, it is type 1. Okay, remember this. And among this one, the most abundant peripheral membrane protein of erythrocyte, it is your spectrin. Okay, and Okay. Now, biochemical test and analytes detected. Okay, we sometimes forget our first year biochemistry practicals, so that's why I'm giving this. So, vouchered test, they can ask you about voucher. See, in some years ago, they asked about this reaction, right? Hmm? Where proline produces yellow color. Okay, so that reaction they asked. So many students were unaware of that reaction. So that's why I'm giving this. Uh, Barfoot's test is for monosaccharide, Biuret is for amino acids. Leave this. This is very important. Early childhood test. Okay, let me go only important thing. Early childhood test is for urobilinogen. Okay, and voucher test is for bilirubin in the urine. Okay, Gerhardt's test, Rothra's test are for ketone bodies. Okay, this one, mean hydrogen reaction. Okay, proteins with minimum of 2 peptide bonds. Sagaguchi test is for arginine. Selivanov's is for fructose. Vandenberg, it differentiate unconjugated bilirubin from conjugated bilirubin. Okay, so with the minimum time, let's go to the some image-based question. Okay, similar to this was there in the previous exam. Look at the this one, and which is the bond marked by the red thing. Which is the bond? Can anyone say which is the bond is this? See, the bond is between a sugar and a base. So this is a beta N glycosidic bond. So this is the beta N glycosidic bond. What is this bond? These are the bonds. What about these bonds? These are the phosphoanhydride bond. So these are the high energy bond. Okay, this is not a high energy bond. Okay, in ATP you have only two high energy phosphate bond. Okay, which is the nature of this inhibitor? Look at this and tell me. So what is happening? Vmax is same. So Vmax is same, but Km is altered. When this is minus 1 by Km, right? That means Km is increased. So this is competitive. So when it is in the V shape, remember like this, okay? So this is just a shortcut I'm telling. Just if it is in the V shape, it will be your non-competitive. Okay, sigmoid, when something is sigmoid in that, it is allosteric. Okay, okay, cooperativity, because of the cooperativity you have the sigmoid curve. Interpret this densitometric pattern of plasma proteins. See this, what is happening? Albumin is decreased, alpha 2 macroglobulin is increased. Okay, so obviously, so this means what is happening? So albumin is excreted in the urine. That means proteinuria, massive proteinuria is happening. So this is nephrotic syndrome. Okay, so what will happen in hepatic cirrhosis? Hepatic cirrhosis also, this will be decreased, but everything will be decreased. This also will be decreased. In multiple myeloma, what will you get? You will get a sharp spike, uh, spike, sharp spike in the gamma region. Okay, so in the gamma region, you will get a single sharp spike. What will happen about chronic inflammation? Leave this acute inflammation. What will happen in the chronic inflammation? You will have a diffuse in the gamma region. Okay, so gamma region, there will be diffuse. Okay, in multiple myeloma, it is just more spike. Why? Because one type of immunoglobulin is produced. It's a monoclonal gammapathy. But in chronic inflammation, it is polyclonal gammapathy. So you get a diffuse increase. Okay, so in acute inflammation, what will happen? You have acute phase reactants. Okay, acute phase reactants, they are in the alpha 2. So everything will be normal. Okay, you will have this one alpha 1 antitrypsin. So alpha 1 you will increase. So alpha 1 antitrypsin C reactive protein. So they come in this region, alpha 1 region. So you'll have that increase also, and some increase in the gamma. Okay, deficiency of which of the following vitamin will lead to this manifestation? So look at this. So it's very easy. You all know it is riboflavin. Okay, so there is something known as castle necklace appearance, right? So that is for pellagra. So that's also typical question, image-based question. They can just give a castle necklace pattern for pellagra. This one, this absorption spectrum belongs to, you can see it is the Soret band. Okay, so we are almost in the time. uh, One student asked this question. Let me answer this question before closing. So he's asking, sir, please tell me, at least, uh, where should I study? Hmm? By making questions from books or by ourselves? So I will tell you, always go for the previous year question. Okay, that is very, very important. Look at the previous year question. So you will always have the advantage. Okay, when you go through the previous year question, you surely will have advantage. And standard textbook question. Okay, if you see these days, they are picking up questions given from the standard textbook. All your standard textbooks are nowadays coming with the self-assessment question. So go with the that questions also. Okay. Hmm. So that's very, very important. And that's what, don't complicate things. That's what I see many students. They like complicating. See, we are not going to penalize you in the exam. Our purpose is simple. We just want you to test your basic knowledge. Okay. So we won't confuse you very much in the exam. The questions will be always like, for example, one question, right? Which of the following restriction enzyme? For example, they ask hill 3, which is the restriction site. This students, some students were there, they were actually memorizing the restriction site. The purpose of the question is not that. Okay, the purpose of the question is you should know that which is a palindromic sequence. Okay, we just want you to know that palindromic sequence is recognized by the restriction and the nucleus. So see among the options, just select the palindromic sequence, you will get the answer. Clear, right? So this is one example I'm giving you. Many students, they're just like in the stress and all, they just confuse things. Relax, cool down. Okay, the exam will be easy. Okay, mainly basics will be tested. So go through the previous year question, go through the questions in the standard textbooks, or you can also make your own questions. It's fine. So it's very happy. I got it's indeed a great pleasure that you all join in this session. Don't forget to attend tomorrow's session also. You will get benefited really. Okay. So I hope you all got benefited from today's session. It's a great pleasure. Good night, everyone. Attend tomorrow's session. Bye bye.